So good morning, good afternoon, and good evening, everyone from where you're joining us today. My name is Nicole Mulaku, and I'm the Program Officer in charge of Library and Consortia here at the Training Center in Communication. And today, together with our partner, Scifiniti Publishing, we are excited to have you all in for today's webinar on research integrity and publishing best practices in biomedical and pharmaceutical research. So I'll be your moderator for the day. And as you can see, I'm sharing a announcement visual of the speakers that we have in line for today's webinar. So let me just quickly take you through the program of the day so that you're all aware. So this is the program of the day. We're going to have opening remarks from TCC by Mr. Owango, who's the Executive Director. And then next will be Mr. Qasit Malik, who's the Publishing Director at Scifiniti. He's also going to give his opening remarks representing Scifiniti. And then our first speaker will be Dr. Kibet from IAVI. And his presentation will be on reproducibility, open science, and research integrity in computation research. Then we have Editor-in-Chief Evidence Synthesis in Healthcare Connect, Dr. Syed, Hasan. And then next we have Dr. James Ayieko from Cambridge, and he's going to be talking about research misconduct, fabrication, falsification, and plagiarism. Then next we have Ms. Noureen Azher from Scifiniti Publishing, and she's a senior editorial manager. And she's going to be giving us a bit of behind the scenes on how publishing workflow safeguards research integrity. And then our last speaker for the day will be Dr. Muhanad, who is the Editor-in-Chief, Biomaterials Connect, also from Scifiniti. And he's going to be talking about research integrity and publishing practices, best practices in biomedical and pharmaceutical research. So all our speakers are here with us today. And feel free to ask your questions that you'll have for them either in the chat, but if you wish to unmute, you can raise your hand if it's a very burning question. But aside from that, we do have the interactive discussion session, and it's also a question and answer session. So you can collect your questions and ask them afterwards, or you can just ask them in the chat and we will address them to the best way we can. So that's the program of the day. Aside from that, just to lay out a bit of house rules, please make sure that your microphones and videos are off unless asked otherwise, so as not to interfere with network connectivity of fellow colleagues who are in the room. And if you need anything, assistance with anything, please let me know in the chat and I'll be happy to assist you. So without much further ado, I'd like to welcome Ms. Joy Owango, the Executive Director of the Training Center in Communication, to give her opening remarks. Welcome, Ms. Joy.
Thank you so much, Nicole, for the kind introduction. And welcome, colleagues. I can see from the chat that some of you are coming in from the US, some of you coming from Asia, the Middle East and Africa. And I really appreciate the time that you've taken to come and listen to this webinar. So regardless of which part of the world you are, it will either be a morning, afternoon, or good evening to all of you. And on behalf of the Training Center in Communication, TCC Africa, it is my pleasure to warmly welcome you to today's webinar on research integrity and publishing best practices in biomedical and pharmaceutical research. We are delighted to host this session in partnership with Scifiniti Publishing as part of our continued efforts to strengthen scholarly communication and responsible research practices across the global research community. This webinar builds on the success of our first session, which brought together a diverse audience from Africa, Asia, the Middle East, and South Asia. The strong participation and engagement we witnessed during that event highlighted a growing demand for capacity building around scholarly publishing, particularly in helping researchers better understand the editorial and publishing processes that shape the global research landscape. Importantly, past webinar we conducted with our partners, Scifiniti Publishing, demonstrated how meaningful dialogue between publishers, editors, and researchers can strengthen transparency, trust in the scholarly publishing ecosystem. We also observed increased engagement following that session, reinforcing the importance of sustained conversations rather than one-off engagements. Today's webinar takes this conversation a step further by focusing specifically on research integrity. Excuse me, colleagues, on research integrity and publishing best practices in biomedical and pharmaceutical research. And that is why we have researchers in pharmaceutical and biomedical research. One of them is from an international research organization, and the other one is from the National Research Organization in Kenya, the Kenya Medical Research Organization. Excuse me, colleagues. So with these two amazing speakers, we are going to have an overview of how we need to have best practices in publishing, especially in biomedical and pharmaceutical research, when you're looking from a national perspective and also from an international organizational perspective. Now, these fields on biomedical and pharmaceutical research carries particularly high ethical responsibilities due to their direct impact on human health and global scientific advancement. So our aim today is therefore not only to raise awareness, but also to provide practical insights into editorial expectations, back-end publishing workflows, and the safeguards that help maintain research integrity in this particular research field. By doing so, we hope to strengthen understanding between researchers and publishers while supporting the responsible growth of scientific contributions from the Global South. We are grateful to our partners at Scifiniti Publishing and to our distinguished speakers for contributing their expertise to this important conversation. Thank you all for taking the time to join us today, and we look forward to a very insightful, engaging session. Over to you, Nicole.
Thank you so much, Ms. Joy, for the kind remarks. And I believe we've all gotten the message to this partnership between TCC Africa and Scifiniti and what we are trying to drive home. And I think this is a good next step to invite Mr. Qasit Malik, the publishing director from Scifiniti, to also give his view on today's webinar and our continued partnership. Welcome, Mr. Qasit.
Thank you, Nicole. Can I share a few slides?
Yes, please,
quickly?
Go ahead. Yes.
Can you see the slides?
Yes.
Yeah. Thank you, Nicole, and thank you, Joy, for outlining the structure of the webinar and shows the importance of this webinar. First of all, I would like to introduce myself. My name is Qasit Malik. I am working as a publishing director at Scifiniti, which is a STM publisher based in Abu Dhabi. My role in Scifiniti is to look after the entire publishing process. Started from the journal launch, the editorial board management, peer review process, and finally the rigorous publication, which is our product. So going forward, let me tell you more about Scifiniti, and I would like to give you the quick snapshot. We launched Scifiniti back in December 2023. We saw a clear regional gap, especially for this region, I mean the Middle East, Africa, and South Asia region. We see that researchers in this region face limited support, fewer publishing options, language barriers, and they have lower visibility in major journals. So our mission is to reduce barriers in publishing while maintaining the international standards. So if you see in just 2.5 years, what we did, we launched 15 journals and more than 450 editorial board members across the globe. We have published 160 manuscripts till date out of 650 submitted manuscripts. We continue to build affiliations and partnerships, and we actively support different conferences, and you can see the training programs like webinars and workshops. So this is all about the Scifiniti in a nutshell. The most important, we have been recognized by the Ministry of Economy last year as a Future 50 Emirati enterprise, and that's really an achievement for us. Yeah, regarding this webinar, I want to have a few words. In fact, I've been in this field in this publishing field from the last 25 years, and I saw that transition. I mean, publishing today is far more different than what we have been seen in the last years. So I think we at Scifiniti think that these sort of webinars and training programs are really helpful especially for the young researchers who wants to know that what are the red flags, what are the best publishing practices. So I'm sure the great speakers will tell you what are the do's and what are the don'ts. So this is the second webinar of our series. The first webinar, that was the general one, we focused on research and integrity, but today we are more focused on red flags and best practices especially in biomedical and pharmaceutical research. That's all from my side, and I really want to thank you to especially the TCC team who worked hard for this, and all the speakers, researchers, students who are attending this session and made this discussion possible. Thank you so much. Good day.
Thank you so much, Mr. Qasit, for your remarks. And I believe we've all gotten now a bit of a detailed analysis brief of our partnership and what we are trying to do. And it is true that over the years, trends in publishing change and everything is changed. So we have to adapt and find new ways to talk about it. So our speaker of the day, our first speaker of the day is Dr. Kibet from IAVI. He's a bioinformatician and an open science advocate, and he'll be talking on reproducibility, open science, and research integrity in computation research. Welcome, Dr. Caleb. Are you on mute?
Yeah,
Okay.
thank you. Thank you so much for the kind introduction, and thank you all for joining even this conversation. So yes, I'm based out of IAVI. We are a vaccine development organization focused on HIV, TB, and emerging infectious diseases. And I'll be focusing my talk on this aspect of reproducibility and open science, especially around this space. So just first is just an appreciation to our funders. And I'll start by this kind of image, and this is a common reality, and it's been a reality for some time. But one thing that we are glad is that this is changing. But it is the aspect of what you come across an article, quite the abstract. It's quite interesting. Maybe it would address a question that you had, but then it's locked behind the paywall. And the other aspect is data, whereby all you get to see is a visualization that might not even present the full picture of the data that you're looking at. So what's the data that was used to produce this visualization is not being shared. So science was and has not been doing so well, but this has been changing, whereby the studies are not replicable. So lack of replication of the studies, low statistical power, which means the number of participants and so forth are not sufficient. P-hacking, so just focusing on trying to repeat this analysis until you produce a p-value that is significant. And then the lack of data publication bias and paywalls. And the other aspect is hacking. So you hypothesize after you know the results. So you analyze, you get the results, and then you create an hypothesis that fits the results that you are seeing. And this all, of course, leads to, with the combination of all these other aspects, leads to a lack of trust from the public. That's Science 1.0, where you gather your data privately, you write your journal article, submit for peer review, the peer review gatekeepers do the publish or reject, and then information is available to the public or not, depending on where you've published and even the accessibility of the information that you've written. The other aspect is Science 2.0, where you pre-register studies, you share ideas, methods, protocol, and data via blogs. So this is accessible to a wider audience. And then you submit preprints, you publish in blogs, you have wikis and in journals. Information and data is available to the public in this case. So that's Science 2.0. And this is science that we are interested in that goes beyond just the traditional publication. And that's open science, the aspect of the practice of science in such a way that others can collaborate with you. So even in the whole process of conducting research, you have collaboration. You have continued contribution by others, and where research data specifically and lab notes and all the research processes that produce your research output are freely available. And these are also available in terms that enable reuse, redistribution, and reproduction of the research and its underlying data and methods. So it goes beyond just the publication itself, but you also have additional contribution, data, methods, protocols, and so forth, which are scientific contributions made by researchers. So a published article is just the tip of the iceberg. It's just the advertising, especially when you're dealing with computational research. That is just the advertising. The real scholarship, the real scholarship is that full software environment, the code, the data that you use to produce these results. So you have to be able to share that full landscape. So if you look at it from the advertising, the lowest scale, which is what most stop at, is just the text. That's just like 0% of really what's considered to be science. But if you bring in data, you bring in the code, you bring in version control, provenance of your data, provenance of the software environment that you're using, and of course, even the full software environment, how to set up the environment to reanalyze or reproduce what someone has done. So reproducibility, being able to redo the study and being able to produce the same research output. That's what's really considered to be science. And therefore, the code is the scholarship. So whereby if someone comes in and says, hey, you really did some great work here, and I really want to use this software that you use or the analysis method that you use for my work. And all they'll just do is, hey, grab the code from GitHub or any other platform. But the other side of it is that someone would be really fearful that someone wants to look at their messy code. What this means is that by being open, you are able to write your code, prepare your data in such a way that it is well organized and clean and descriptive and so forth. So there is a benefit of being open. So the traditional research and publication was whereby what the researcher does remains with the researcher. So the whole walls of academia, whereby you have the lab work, the validation studies, the data, the paper, the thesis, the re-experiments that you did, the rewrites that you did, all those are not accessible to anyone else other than the researcher. And if you move it out, you have the publication, whereby you have the publish, that publication belonging to the publication process belongs to the publisher. The output as well belongs to the publisher. But what we want is to break these walls of academia where the data, because really researchers do a lot, but what's exposed is very little. So going beyond that, you get to see that now data science is becoming central in biomedical sciences, and therefore this practice of open science, collaborative tools need to be adopted in this space. If you look at reproducibility landscape in itself, from the aspect of really well documentation of your research process, RStudio, Jupyter Notebooks, and so forth, and version control, Git and GitHub, and the rest. And then the software environment, so preparing workflows in SnakeMake, or even moving beyond that to preparing containers. So if you think of a container, you're able to move the whole software environment, the data, and someone can be able to quickly reproduce the work that you did fast. And sharing all these, really what it does is it facilitates reproducibility. So going to fair data, so findability, accessibility, interoperability, and reusability of resources, which is in data and software, is in a way that it is shared to enable and support reuse. So the aspect is reuse. We need to be able to stand on the shoulder of each other to move beyond, to move research beyond where we are. And therefore, fair data should be as open, especially when you're dealing with pharmaceutical, biomedical sciences. it should be as open as possible and closed as necessary. So data can still be restricted, but be fair. So there is a process for being able to access it. Controlled access is really the word. And open data is just a subset of all the data that is shared. And what's really important is to protect the privacy of the data subject. So they should remain central to your data sharing. And the whole process stands from being findability, which is the metadata, and then the accessibility, what determines what to share. You don't share everything. You need consent, risk management, and so forth. Interoperability, what are the data standards that you're using, which are accessible to others? And then, of course, the license that you're using to share your data, which provides what's permitted and what's not permitted. So to be fair, you have to really shift your mindset and ensure we have to really look at the incentive structures, because the incentives right now focuses on the publication alone. But really, if we provide incentive to the sharing of data, sharing of code and the rest, then people would be able to put in time and effort to sharing of those. And then there's investment in data management planning, and of course, investment in platforms and structures to facilitate that. And these are some of the things that publishers would be involved in, but also the researchers as well. And so fair data is really controlled access, especially when you're dealing with pharmaceutical science. And ultimately a goal without a plan. So you want really to be fair, but you really need to have a plan. Otherwise it will just be a wish. And therefore that's the need for a data management plan, managing it from the beginning. So a data management plan focuses on describing the data itself that will be authored or generated, how this data will be managed, how do you manage it during the whole process of your research, and ultimately how it will be made accessible throughout its lifetime, and ultimately when you get to publication. So my hope is really that future generations will look at that term open science and reproducibility as a tautology. So it's just a throwback from an era before science work, before science really became science. And open science really will not be talking about open science, but we'll be saying we are conducting science. And of course, these close secretive practices that define our current culture will become primitive, especially to them as alchemy is to us. So thank you so much for your attention and for listening to me. I'll take questions later on. Thanks.
Thank you so much, Dr. Caleb, for that very insightful and detailed presentation. I think you brought the message home, especially I like the aspect on data management, because it's very important that we're able to effectively manage our data and not have it scattered everywhere. So thank you so much for that. So any questions for Dr. Caleb, please put them in the chat. He'll answer them, or he'll answer them also in the question and answer session. So our next presentation is from Dr. Syed Hasan. He's the Editor-in-Chief, Evidence Synthesis in Healthcare Connect from Scifiniti. And he's going to be talking about research integrity and publishing best practices in biomedical and pharmaceutical research, evidence-based research integrity. Welcome, Dr. Syed.
Can you hear me?
Yes, we can hear you. Go ahead,
And
sir.
you can see the slides as well.
Yes, sir.
Okay. Thank you very much for the introduction. So I'm Shahzad. I'm the Editor-in-Chief of Evidence Synthesis in Healthcare Connect Journal, and I'm also Associate Professor at the University of Huddersfield in the UK. So these are the objectives of my presentation today. So I will be mainly focusing on the research integrity in evidence synthesis, and use the examples from the literature, the evidence synthesis literature where possible. So research integrity, failures do not mean that it's a deliberate fraud. In many cases, especially in evidence synthesis, they originate the failures, the integrity failures originate from data-related issues or problems, such as poor documentation, poor record keeping, and unclear the design or search strategy in evidence synthesis, or improper handling of figures in data set. So this figure is from the the latest comprehensive review of retractions of medical literature from 1975 to 2024. And this summarizes the leading causes of the retractions in medical literature or publications. And the majority of the cases you see is related to the data consents, which almost accounting more than quarter of the retractions. And these concerns often involve issues like inconsistent data sets, duplicated data sets, or manipulated images, or missing underlying data. And another interesting finding is the year 2020, where the COVID-19 happened in the end of 2019, and in 2020 there were lots of publications, especially randomized controlled trials, and later they were retracted many because of the poor design, because of the manipulation in the data sets, and there were lots of retraction, especially in evidence synthesis as well, because in evidence synthesis, like systematic reviews and meta-analysis, we rely on the data from original studies, from randomized controlled trials. So if something goes wrong with the randomized controlled trials, so ultimately it will impact the evidence synthesis as well. So we have to be very careful when we are dealing with published data from randomized controlled trials, or from the original studies when we are using it in our evidence synthesis. So we have to critically appraise before we use it in our evidence synthesis. So there are other factors as well, impacting the integrity. That includes the fraud, the peer review issues, and ethical violations also contribute to retractions, but they occur less frequently than problems related to the integrity and handling of the research data. So data concerns is the main cause of retractions. So the message is that many of these issues are preventable, and by improving the documentation, by improving the design, the study design, maintaining clear records, and ensuring transparency in data and image reporting. So researchers can significantly reduce this issue of the integrity failures and concerns, especially during the peer review process as well, after the publication. So that's why journals, they are also increasingly apply pre-submission and pre-peer review screening, asking authors to make many changes, especially related to the data, and also looking at the academic misconduct issues, plagiarisms, image analysis, and verifications of ethics compliance. So now if we look at the journal, what they actually expect. So I will speak from my journal's point of view, which is evidence synthesis. So the issues are same that we routinely encounter, and this is what slows down the manuscript processing, because we request, in pre-peer review process, we request information from the authors, and sometimes authors, they fail to provide the especially the information related to the data. So at submission, we expect that the results in the manuscript can be traced back to the original data. So in case of evidence synthesis, we request the search results, the search strategy, and the records from the different databases the authors they extract. So it doesn't mean that authors, they have to upload all the related data immediately, but authors should be able to provide it when requested. We also look for the complete reporting, the search strategy, so they can adhere to the PRISMA checklist, for example, in case of systematic reviews. And incomplete reporting is one of the major reasons reviewers lose confidence in results, because they cannot trace back the data, they cannot see the proper documentation. In some cases, in lab-based research or original research, there is an issue of image integrity as well. There is, this is some manipulation with the data, and there's another issue. And third is in all these submissions, in our journals, they go through peer review screening. So this can include similarity checks, figure screening, and ethics and compliance verification when relevant. If these elements are missing or unclear, the paper may be sent back for clarification before it even reaches the reviewers, just to maintain the integrity of the scientific record and save time later. So the message is, when authors are transparent from the start, review process moves faster and the risk of corrections or concerns after publication drops significantly. So from this figure, it's obvious that the poor methodology produces biased evidence, and selective reporting, reproducible, unreliable research, and flawed study distort the scientific record. Like in case of COVID-19, there's a lot of retractions during that time because of the flawed methodology, unreliable results, and the issues related to the data and the scientific record. And that actually misguide the clinical decision making. Because in healthcare is very critical, because any healthcare professionals, they can use your evidence, your article, to change any drug therapy for their patients. They can use it in their clinical practice. So this decision making, that the flawed methodology or the poor scientific evidence can impact or misguide the clinical decision making. So biased research finding influence the funding priorities as well, policy decisions, and patients' care as well. The weak method therefore lead to incorrect clinical actions and harm the quality and safety of the healthcare. And we can easily see that immediately after submission, pre-checks are run. And if the integrity matters are addressed, the workflow proceeds without any back and forth revisions. If they follow the checklist in case of evidence synthesis, is easy for the journal, and also for the authors. They can add any missing information or critical information in their manuscript. So here is a simple way to think about rigor. So it means it includes the design, not only design, plus the implementation or execution, plus the reporting of the results, so that others, the readers, they can judge the work and reproduce it, the reliability. We always talk about accuracy and the validity and reliability, which is accuracy and reproducibility. So you have to be clear in your documentation, so when other people, they are reading your manuscript or your article, they can reproduce it. That is the reliability of your work. So first is the design. So this design is really important. And in evidence synthesis, our design, we are not doing any original research. So we are relying on original research in evidence synthesis. So we are reading published papers, published data. So we have to be extra careful when we are looking at the original research and the design. And sometimes you're only interested in one particular design, like a randomized controlled trial. So we have to look at all the elements of randomized controlled trials, critically evaluate all the steps involved in the research, and then we include it in our evidence synthesis, so to avoid any retractions later. So we look at the clear hypothesis, appropriate methodology, sample size, the proper controls is very important. Sometimes in randomized control trials, they don't have the same control, comparable control in their studies. So we have to look at the controls as well. And these elements, if we check carefully, reduces the bias and strengthen the credibility of yours in evidence synthesis. Second is the execution. Even a strong design can fail if execution is not controlled. So we look for validated protocols. That's why, in clinical, for randomized control trials, they register their trial and the protocols are available on different databases. So you can look at their protocols, the compliance issues. Sometimes they deviate from their protocol. Sometimes they don't share their data. So quality control steps and statistics also, we check whether it's aligned with the research design. Third is the reporting. In case of evidence synthesis, we use a standard checklist for reporting. So there are two types of checklist in evidence synthesis. One, we use it for reporting the results, and one is for the critical appraisal of the included studies. So a lot of time authors, they use reporting guidelines for critical appraisal. So there are separate guidelines for critical appraisal of the included studies, and there are separate guidelines for or checklist for the reporting of the study results. So a quick practical point is, I would recommend using the the reporting guidelines, adhere to the guidelines. In case of evidence synthesis, a majority of the people, they use Prisma checklist, Prisma guidance for completing the evidence synthesis. I have mentioned about reproducibility and transparency. So the goal is simple. We have to make it easy for someone else to verify what we have done. So in practice, the good evidence, the good piece or the good article looks like their methods are detailed enough that another person or the researchers could repeat the experiment or the process like in evidence synthesis without guessing. So key resources are precisely identified, so there is no confusion about search strategy or your keywords that you have used, the different databases you have used for synthesizing the evidence. So a logical sequence will help you define the purpose, scope, and design of your evidence synthesis before searching the literature. So that's why it's very important for us to publish the protocol on databases such as Prospero. So we can publish our protocol, we can define our purpose, scope, and also the search strategy, and we can publish it on Prospero or other databases, so that you can see the compliance as well, and you don't deviate from your original plan. A common integrity issue is when the claims don't match the data. So reviewers, they can often sense this when the narrative feels too perfect or when outcomes are inconsistent across sections. So being transparent about what was planned versus the exploratory analysis build the trust. So in case of evidence synthesis, I already mentioned that we encourage the authors to publish their protocol with Prospero, for compliance and for proper documentation. We expect authors to provide the full search strings or search strategy, so readers can see the exact search logic used in each database and verify how studies were identified. We also ask author to put all the filters they have used in finding the relevant literature. We also, expect authors to provide the search histories or the search results in supplementary file. They can include and provide a clear record of the search process for documentation, verification, and future updates, and report the search date so readers can see. Sometimes, when you're looking at the evidence synthesis of prevalence studies where you're giving the percentage of, for example, of diabetes in a particular region or global estimates, it's very common now. You report like global burden of disease, they report percentages of people with certain diseases. So you cannot include, but some of the recent systematic reviews published in very good journals, they included records from 1990s or 1980s, which is like 30 years ago. And you are presenting the prevalence estimates and saying that the prevalence of this particular disease is 20%. In 2025 or 2026, you are reporting, and you are relying on data which was published in 1995. So this is not possible. This is not the appropriate, approach. But you see that many good journals, they don't look at, the dates they have used or the data they have used, from, the year they have used or included in their search. So it's very important that you depends on the purpose as well. If you're looking at prevalence, so we are interested in the latest estimates, recent estimates, and then you use and rely on recent articles that reported the prevalence, not like 30 years ago or 40 years ago. And then we also enable replication by providing sufficient methodology details to enable the search strategy to be repeated in verified. So this is different from other research. In other research, they have the original data, the authors, they generate the data. But in case of evidence synthesis, we rely on and publish data from somebody else. So in our case, search strategy, keywords, the protocol, these steps are important for documentation and for compliance. So now, for the journal's point of view, a lot of authors, they have to wait, very long to get their articles published, and it's mainly related to the ethics and compliance checks. So in many cases, papers, they don't get delayed because science is weak. Scientific part is very strong. They get delayed because key ethical or information issues are missing or unclear. From an editorial perspective at Scifiniti, we often see a predictable set of missing pieces that trigger queries before or during peer review. See, on the author side for human studies, the basic must be clearly stated. Ethics approval, for example, is very important. So it has to be included in the research or in the manuscript. And the consent, patient consent is also important. Data handling, privacy, and confidentiality issues are also important and should be disclosed. There are other requirements for clinical trials. The authors, they check for the trial registration, so they can check, they can provide the registration detail as well. On the journals verification side, editors, they commonly verify conflict of interest as well, which is important, especially if they are working for any pharmaceutical industry and they're writing for a particular pharmaceutical product and they have conflict of interest because maybe they are promoting their own product, and sometimes they don't publish or disclose the complete data set, especially related to the adverse effect of any new medications. So that's the leading cause of concern in when we are handling clinical trials or any randomized controlled trials. So there is an issue related to the conflict of interest. Sometimes it's financial, sometimes it's not financial. So we also check for authorship clarity. So there is a contribution statement we ask author to include based on their contribution. Gift authorships or ghost authorships are very common. So we have to check the contribution and authors to provide that in some detail. Another issue is AI, but I will discuss that in later in next slide. So the takeaway ethics and compliance statements are not administrative extras. They are part of research integrity. And getting them right upfront is one of the fastest ways to avoid delays and keep our manuscript moving smoothly. So now I want to focus on a few important issues that are related to integrity. So from Scifiniti point of view, the editorial perspective, these are the patterns that most often trigger peer review queries, reviewers' concerns, or any integrity check. First is the plagiarism or text recycling. This includes high similarity. So all the submissions, they check, the similarity, and they provide the reports to the editor. They check, and editorial staff also carefully check all the submissions for any suspected plagiarism or text recycling. Second is the image manipulation. It's not common in evidence synthesis, but in original research, lab-based research, the image manipulation and duplication is very common. And then the data inconsistencies, things like impossible values, mismatched sample size across sections, or claims that do not align with the underlying data. We also sometimes see paper mill signals such as template style text. So if they have the template, they know that they have this, they develop this skill, and then they start writing, 10 and hundreds of papers, on the same template or style text. Then there are ethics gaps. In the original research mainly, it's related to ethical issues and the compliance. And then the peer review manipulation, for example, suspicious reviewer emails or attempts to influence the citation. Sometimes, reviewers, they have to look at carefully, because sometimes when you get the report and then the reviewers asking authors to include their own, their references or citations or articles, so pushing for that so that you include. Sometimes it's not relevant, your topic is different, but they're asking authors to include, the certain or their own citations or articles in their manuscript. So the purpose is, of this slide is not to alarm anyone, but it's to show how and where delays and rejections often come from. If authors address these areas early, papers move faster and the final publication is stronger. So this is a checklist for authors and reviewers, the things that from an editorial standpoint at Scifiniti most consistently help manuscript move smoothly through screening and peer review. First is before we check, for similarity. Second, we confirm the ethics and consent and registration. Then, the figures and the data and the methods is reproducible, reliable methods. And fifthly, align the data availability with your claims. And if editor raises any integrity issue, respond with evidence, not just reassurance. Provide raw data or raw, files or logs or clear explanation. So this table presents the current uses of AI in evidence synthesis. Even the big journals like BMJ or Lancet, they are not stopping authors to use AI. What they want is the declaration. You have to declare the use of AI in your manuscript. And your methods, if it's significant, you have to write. You cannot include AI as an author. There, the guidance is very clear. But you can, if you are using it, you have to declare. And then the second thing they highlight or emphasize is the human verification. So you have to verify all the facts and information you are taking with the help of an AI. So this table presents the current uses of AI tools in evidence synthesis. Some of these uses are absolutely fine, but generating the whole manuscript based on AI is not acceptable. So AI supports evidence synthesis by managing large volume of literature. Even there are now platforms like Ryan. Ryan is a platform available which is AI enabled platform for authors where they can handle the large volume of literature and they can automate the whole process. So it accelerates, the title and abstract screening and assisting with search strategy development and data extraction. These functions improve efficiency and help researchers process large data sets that would otherwise require extensive manual effort. So you can automate your process, but you have to report it in your methods if you have used any platforms like Ryan in your evidence synthesis. AI also strengthens the writing and synthesis process by improving language clarity, summarizing complex evidence, and reducing the time pressure associated with academic publishing. This allows researchers to focus more on interpretation, methodological rigor, and critical analysis. So the message is no journal, even the big journals, they are not stopping authors to use it. What they want is the declaration and human verification. You have to disclose it. A lot of people, they fear and they don't disclose the AI use. They are using it, but they are not disclosing it. And when they run the for similarity checks, and then they identify very high use of AI. So it is, create a trust deficit. When the editorial staff or the editors, they're looking at very high percentages of AI or similarity percentages, it's difficult to deal with, that submission and to proceed. So these are the limitations with the current AI tools. They give you a lot of hallucinated citations, false citations. So you have to check. That's why human verification is important. They cannot implement any of the eligibility criteria of your evidence synthesis explicitly. And then there are issues with the training data, over-reliance without verification, and data privacy concerns. So responsible use is when you verify all the extracted data manually, check original articles for accuracy, disclose AI assistance in methods, and use AI as a support, not replacement. And another issue is that most of the AI tools available now is the purpose is, the communication, conversational AI tool, and people, they are not, specialized research tools, and they are likely to introduce inaccuracy, bias, and plagiarism. So finally, I just want to briefly discuss the role of reviewers because maintaining research integrity is a shared responsibility between authors, reviewers, and editors. Reviewers play a key role in identifying issues that may affect the reliability or interpretation of a study. The first thing reviewers should check is whether the research question, study design, conclusions, search strategy, the protocol, actually align. Sometimes manuscript overstate their findings and make claims that go beyond what the data support. A careful comparison between the question, methods, and results and conclusions can quickly reveal this. So reviewers should also look closely at figures and images and the values they included in their figures or tables. If anything seems unusual, such as repeated pattern, possible duplication, it is appropriate to ask the editor, whether the original data or files can be reviewed. So as a reviewer, you can request to editor, and then they can request from the authors to provide the raw data. Another important area is methods and statistics. Reviewers should confirm that the approach is appropriate. The study design and then methods are described clearly enough for someone else to understand or reproduce that work. Ethical compliance is also essential. Reviewers should check whether ethical approval or trial registration or conflict of interest disclosures are clearly stated. So in this way, reviewers can help ensure that the final published work is credible, transparent, and scientifically sound, which ultimately benefits both the research community and the integrity of the research record. So that's all, and thank you very much.
Thank you so much, Dr. Syed, for that detailed presentation. That was amazing. Thank you so much for your time. Quickly, we'll move it over to our next presentation. That's Dr. James, and I will be sharing his presentation for him. Yes. So
Yeah,
welcome, Dr. James.
Yes, thank you. It's an honor to speak to your audience today on research integrity. My talk will focus on research misconduct. Glad to see that Dr. Syed has, given us a great segue into this matter. So I'll focus on falsification, fabrication, and plagiarism. I'm a research scientist at the Kenya Medical Research Institute, by training a medical doctor as well as an epidemiologist with focus on HIV and hypertension. Next slide. So I just want to start off by saying that, integrity in research is like a glass of water. And so once it's broken, it can never be the same again. And so as researchers, you carry the burden and the responsibility for the trustworthiness of your research. And this presentation is just going to focus around the theme of trustworthiness and see implications of not being trustworthy. And so we'll be delving onto two or so case studies to enable us to reflect on research integrity as we do our work, as well as put up our publications and submit them to journals for publication. So essentially, there are 4 principles of research integrity that I'd want us to think about as we go through this presentation. 1 is integrity, 2 is accountability, 3rd is professionalism, and lastly, stewardship. Next slide. So falsification is as what is described in this cartoon, making your data look different from what it actually is. Next slide. So it is the act of manipulation of research materials, equipment, or processes, or changing or omitting data or results, such that your results are not accurately represented in the research record. You kind of spice it up to appear in a certain way. Maybe some of you have been to presentations where the results look too neat, they are too orderly. And it would always arouse your suspicion as to how results look the way they do. So nature has some sort of order, and that's what we try to kind of establish when we conduct research. But it's an order that is not, is not as organized as it would be man-made. So I think that's the concern to think about. The next aspect to talk about is fabrication in the next slide. And here it's about making up data, essentially. So I hope you get the point from this cartoon. People have different interests, but you have to uphold your integrity in terms of outcomes that you're going to share with the global audience in terms of your findings. Next slide. So as I've said, fabrication refers to making up data or results and then recording or reporting them as if they were actual data. Next. Plagiarism, I think, has been described. I won't take too much on it, but in the next slide, we have a short definition of it. It's the act of appropriating someone else's ideas or processes, results, or words without giving appropriate credit. So referencing, citing individuals for their contributions. So at this point, we go into the cases in the next slide. So I'll talk to an example to, speak to one of the examples of what Dr. Syed has just talked about with, we had a lot of publications, between the period of 2019, 2020, when we had COVID, because a lot of, there was gap in terms of understanding COVID. And so researchers went high drive to try and put together evidence to help with treatment. So our first case is about evaluating a study that set out to evaluate the benefits of hydroxychloroquine or chloroquine used for COVID-19 treatment. And so this analysis was led by Dr. Mera from Brigham Women's Hospital. And what he set out to do was just an analysis of data from multinational registries. So researchers were not directly involved in data collection. As you see in the orange box down there, the amount of data that they had was quite massive. 671 hospitals is pretty impressive and admirable for any epidemiologist who wants to conduct a research. And multi-continent, 6 continents, and so they selected dates of review, and then they were looking at patients who are given chloroquine with or without a macrolide and hydroxychloroquine with or without macrolide compared to controls who received none of these drugs. Next slide. Their main outcomes were to review deaths that were reported as well as arrhythmias, those are irregular and heartbeats, and their results showed something that was informative when you have a glance at it, that deaths were lower in controls and arrhythmias were more in chloroquine and hydroxychloroquine and the chloroquine group. That would then suggest that there is harm that these two medications present, and you are better off not taking them. Now, the challenge was that they were unable to their result was that they are unable to confirm that these agents were conferring any benefits when used for COVID-19. So this went on to be published in a high-impact journal on May 22nd, 2022 in the New England Journal of Medicine, which is a decent and pretty prestigious journal. Next slide. So, as you're aware, their results raised alarms on the safety of drugs that have been used for COVID. And so it essentially called for scrutiny of the data that was used. So the researchers, trying to be meticulous about what they had put out, called for scrutiny, as well as others who found these results interesting. But after trying to look through the data that they had, the authors had to yield and retract their article in the New England Journal of Medicine. And, what they said was that we can no longer vouch for the veracity of the primary data sources. And due to this unfortunate development, they request that the paper be retracted. You're aware that they were not collecting data. They were just aggregating data from these 671 hospitals. And because they can't vouch for it, they cannot tell the global audience that this is the position. So the article was retracted on 4th of June, 2020. So this slide just shows the retraction, and it's where they acknowledge the limitation and the fact that they are unable to vouch for the data. So this seemingly important finding had to be withdrawn because it cannot be verified. Next slide. If we had time, I would have asked the audience whether you think it was an issue of falsification or fabrication. But because of time, I won't ask. Maybe something to ponder about. But my consideration on this is that it's potentially a combination of both, or falsification of data that the hospitals may have had. Next. So our second case is, I choose to share these and share names because they are in the public domain and it's a case that is known, but I just highlight it so that we reflect on something that has to do with publication and our opportunity as scientists to review articles from that are sent our way, by journals. So this case is a case of Dr. David Bridges, a professor at that time, that was towards the late 80s, a professor at Bayer College of Medicine. And so a doctor referred to as Robert Randall from Harvard Medical School had submitted a manuscript on their research to this journal. And in short, the work that he had done was, trying to explain vision. Our vision is processed through a film at the back of our eyes, referred to as the retina. And so the science at that point was trying to explain how do our eyes work on reprocessing and creating a new image after looking at one and then moving to a new image. So Randall found out that regeneration that happens in the retina is something to do with isomerization, so changing of the form of the pigments at the back of the retina. And that was groundbreaking. So he sent the article to the journal sent the article to Dr. Bridges, who is, was an expert in that field, and he was asked to review it. It took Dr. Bridges 1 month to give a response, and he returned the paper and saying he was working on the same subject and he couldn't review the article. Now, after publication of his findings, Dr. Randall complained to the NIH, which was the funding agency for his research, because there was striking similarity between what he had submitted to the journal and what Dr. Bridges had published. Next slide. So what NIH proceeded to do was just to set up an inquiry to understand the irregularity, what may have happened with this. And indeed, they confirmed that Dr. Bridges altered the dates on his work to suggest falsely that he had begun the research before reviewing Dr. Randall's work. And he did not acknowledge the source of the information. So the conclusion was that Dr. Bridges had plagiarized Dr. Randall's work. So on the yellowish box to the right, I just wanted to focus on the implications of these kind of things and take you back to my example of the glass at the beginning of the talk, that after working on our careers for a long time, they can go down, in an instance because of lack of trustworthiness or integrity. So the recommended sanctions by NIH were article withdrawal, and current NIH grants that he had were also withdrawn. He was prohibited from federal grants, and then also excluded from the peer review programs for 10 years. And, as you could see from the picture, Dr. Bridges was, a full professor, advanced in age towards the tail end of his career. You can imagine what image he may have left his career with. So I think that's my last slide because of time, but this article is out there in the public, and you can take a look at it on the vision paper. That's my last slide. But I think just to reiterate on the 4 principles of integrity, you want to think about trustworthiness, being honest in your work, being accountable to whoever it is that you are communicating to, as well as the people you've collected data from, professionalism and stewardship of science. Thank you for your attention.
Thank you so much, Dr. James, for that presentation. And thank you for sharing with us your thoughts on this topic. Our next speaker of the day is Miss Noureen Azher. She's the Senior Editorial Manager from Scifiniti, and she's going to be talking about behind the scenes, how publishing workflows safeguard research integrity. Welcome, Miss Noureen.
Thank you, and hello, everyone. Thank you for joining our session today. Let me first introduce myself. I am Noureen, and I am working as Senior Editorial Manager in Scifiniti Publishing. And I have been in publishing industry for over 17 years, and I was previously working for Bentham Science Publishers. Let's move on our topic, which is about safeguarding research integrity and how Scifiniti Publishing works to maintain the integrity of the research we publish. So today I will focus here on 2 areas, which are about publishing workflows and research integrity and ethical compliance checks, which we perform at different stages of our publication cycle. I just want to emphasize here that all our publishing practices are in accordance with the COAP guideline and ICMJ and other international publishing standards. So now let's focus on the first topic, which is about publishing workflow. So whenever we receive a manuscript, there are different stages at which we perform our ethical and integrity checks to make sure that the research which we publish is in accordance with the international standards. So first, when a manuscript is received, author during the submission processes are required to disclose any kind of conflict of interest. So we can take care of that during the processing of the manuscript. Once the manuscript is received, we performed initial screening that comprises of two types of checks. The first one is about the technical checks. Another one, which is the most important, is about ethical and integrity checks. I will discuss this in detail in my next slides. But in the next stage, when a manuscript is successfully passed through these checks, it's forwarded to the editor-in-chief for the initial decision. But here again, we make sure that the EIC do not have any conflict of interest with the author. And in case EIC has any conflict of interest, then we assign that manuscript to any other editor of the editorial board, and instead of assigning it to the EIC. Once an initial decision is taken either by the EIC or by any other editor, manuscript is proceeded to the peer review process, where again we make sure that reviewers which are assigned to the manuscript have no conflict of interest with any of the author listed in the manuscript. So make sure that the review process is transparent and unbiased. Every version of the manuscript which we receive, we make sure that they pass through the same ethical and integrity checks. So once we receive a revised version, it passed through the same integrity checks, and the final check is being done after the EIC decision, after which the manuscript is published online. And even after the publication of the manuscript, there are some concerns from readers or any other, then those concerns are resolved in accordance with the guidelines given by the COPE and in other international bodies. So this is the second point, which is about the key checkpoints which help us to maintain the research integrity. Here I will discuss some of the points, although they are discussed in detail by Dr. Hasan, but I will discuss here how Scifiniti team works in coordination with our editor-in-chiefs to make sure that what we are publishing is in accordance with the research integrity. The first point is about the plagiarism prevention. The second one is AI usage. Third, authorship criteria. And then we have ethical approvals and reporting guidelines. Then there is citation manipulation. And the last one is conflict of interest. So for plagiarism screening, when a manuscript is received, is passed through the cross-step similarity check. But we make sure that whatever the report is received is analyzed by any team member, and decision is being made on the basis of human judgment. So on the basis of the decision, manuscript can either be revised, rejected, or author are recommended to rephrase the manuscript, or they are also recommended to cite the references. And whatever decision we make during this process and all other checkpoints, they are done by taking input from our editor-in-chiefs. The next point is about the AI detection. Here again, we process the manuscript through AI detection tool, a service which we have taken from the Crossref. And after receiving the report, it analyzed by a team member, and whatever the analysis is being discussed with the editor-in-chief, and based on the decision of the editor-in-chief, we recommend author about either to revise the manuscript. They can either name the tool they have used and the reason why the tool has been used. And this AI detection or AI usage is not limited only to authors, but our editors and reviewers are also recommended to make sure that during the review process, they do not upload manuscript to any AI tool, and the reports which they are preparing are based on their judgment instead of getting the feedback from any AI tool. If, however, they find any kind of AI content, then they may contact the editorial office, which will coordinate with the author to resolve the issue. Next point is about the authorship. So when a manuscript is received, we do verify all the authors and make sure that all the authors listed in the manuscript have their contribution in accordance with the ICMJE authorship criteria or credit rule. We do not allow gift, ghost, guest authorship, and also AI authorship is not allowed. However, these non-author contribution can be listed under acknowledgement sections. We do perform checks for author verification, their credentials, their affiliations, their email addresses, their previous publication records are being checked. For students and independent author, we have some additional checks which we performed, and any kind of changes in the authorship deal in accordance with the COPE guidelines. Either they want to remove any author or they want to add any author, then authors are requested to provide the reason, their contribution, and then we make changes as per the guideline of COPE. Next point is about the ethical approval and reporting guidelines. So based on the article type, authors are required to provide approval from an ethics committee if it is applicable on their research. For example, if they have worked on human, then they are required to mention this that they have followed Declaration of Helsinki while preparing the manuscript. For animal research, they are required to prepare the manuscript according to ARRIVE guidelines. For consent to publication and consent to participate is also required in each of the manuscript where it is applicable. Based on different article types, some other guidelines are recommended, and authors are required to follow those guidelines while preparing the manuscript, and they are also required to declare those guidelines in the manuscript. These different guidelines are PRISMA, CARE, Trends, and some other which are already listed on our website. Next point is about citation manipulation. We highly recommend our authors to cite only those references in their manuscript which are relevant to the topic, and citation should not be done just to increase the citation count. So we highly recommend author and self-cited references are being checked by our team to make sure that these are relevant. In addition to the author, reviewers and editors are also recommended to refer only those papers which are relevant, and this reference citation should not be done to manipulate or to increase the citation count. The next point is about the conflict of interest. As I mentioned in my first slide, that during the submission process, authors are required to mention if there is any personal, financial, or professional conflict of interest, and they are required to mention this under a section which is the conflict of interest. So we can take care of these things during the processing of the manuscript. Similarly, for editors and reviewers, we make sure that they do not have any conflict of interest with the author, and we also recommend them to disclose if there is any conflict of interest. And if there is any conflict of interest which is not disclosed and we later on find out, we deal all those cases with the help of COPE guidelines. So there are some other text points, but I have discussed here some of them, and I hope that you are able to see that how Scifiniti works at backend to maintain the research integrity. Thank you so much.
Thank you, Ms. Noureen, for your presentation. I really appreciate you taking the time, and thank you for the insights you've given our audience today. So our last speaker of the day is Dr. Muhanad. He's the editor-in-chief by Materials Connect Scifiniti, and he's going to talk about research integrity and publishing practices in biomedical pharmaceutical research. Welcome, Dr. Muhanad.
Hi, Nicole. Thank you very much for your introduction, and thank you very much for inviting me to be here in this webinar. I will start sharing my presentation. Can you see my presentation?
Yes, you can. Go ahead.
So the title of my presentation is Research Integrity and Publishing Best Practices in Biomedical and Pharmaceutical Research. First of all, I would like to thank Malik Qasit for helping in putting together this slideshow. My role is editor-in-chief for Biomaterials Connect for the past 2 years, and along my academic role at Georgia University of Science and Technology as an associate professor. So our agenda for today will be talking about why experimental integrity matters, data and image integrity expectations, methodological rigor and reproducibility, ethics and compliance checks, common integrity issues in biomedical pharma submissions, and practical editorial expectations for authors and reviewers. So hopefully I won't take long, but I'll try to touch base on these aspects from my background as editor-in-chief in Biomaterials Connect. Integrity failures often originate from data-related problems, not only deliberate fraud. Experimental integrity is so important in biomedical and pharmaceutical research. Integrity failures do not only arise from deliberate fraud. In many cases, they originate from data-related problems, such as poor documentation, unclear experimental procedures, or improper handling of figures and data sets. If you look at this chart on the right, this has been taken from one of the latest researches conducted in Turkey. It summarizes leading causes of retractions in medical publications. We see that data concerns account for the largest proportion, over 31% of retracted papers. These concerns often involve issues like inconsistent data sets, duplicated or manipulated images, or missing underlying data. Other factors such as fraud, peer review issues, and ethical violations also contribute to the retractions, but they occur less frequently than problems related to the integrity and handling of research data. The key message here is that many of these issues are preventable. The question is how we can prevent them. Simply by strengthening experimental design, maintaining clear records, and ensuring transparency in data and image reporting. Researchers can significantly reduce the likelihood of integrity concerns during peer review or after publication. This is also why journals increasingly apply pre-submission and pre-peer review screening, including plagiarism checks, image analysis, and ethics compliance verification. With this context in mind, I will now move to the next topic, data and image integrity expectations in submitted manuscript. Now that we have seen why integrity matters, let's translate into what journals actually expect. And I'll speak from the Scifiniti Publishing and Biomaterials Connect journal perspective, because these are the same issues that we routinely encounter, and this is what slows manuscripts processing down. First is data integrity. At submission, we expect that results in the manuscript can be traced back to original raw data. That does not mean everything must be uploaded immediately, but authors should be able to provide with it when requested. We also look for complete reporting like sample size, replicates, exclusions, and clear statistics, because incomplete reporting is one of the most common reasons reviewers lose confidence in results. Second is image integrity, which is a frequent pain point in biomedical submissions. Some adjustments are allowed, like brightness or contrast, but they must be applied uniformly and must not change the scientific meaning. Problem typically arise with selective enhancement, undisclosed splicing or in gels plots, duplication or unclear labeling. From an editorial standpoint, we also expect authors to retain the original unprocessed images, because that's what resolves the questions quickly if concerns come up. Third one, and this is important, at Scifiniti, all submissions go through pre-peer review screening. This can include similarity checks, figure screening, and ethics compliance verification when relevant. If these elements are missing or unclear, the paper may be sent back for clarifications before it even reaches reviewers, just to maintain integrity of scientific records and save time later. So the key message is simple. When authors are transparent from the start, review moves faster, and the risk of corrections or concerns after publications drops significantly. If we look at this flow chart, we can easily understand that just after submission, pre-checks are run, and this is the step where, if integrity matters are addressed, then the further workflow proceeds smoothly without any delays and without any back and forth revisions. Here's a simple way to think about rigor. Rigor equals design plus execution plus reporting so others can judge and reproduce. First, design. In terms of design, reviewers at Scifiniti typically look for clear hypotheses and appropriate methodology, including sample size, proper controls, and etc. These elements reduce bias and strengthen credibility before we even look at results. Second is execution. Even a strong design can fail if the execution is not controlled. So we look for validated protocols, quality control steps, and statistics that match the research design. Third is reporting. This is where many otherwise good studies lose time. The methods need to be complete, exclusions must be transparent, and authors should use guideline checklists when applicable. If protocols are available, even as supplementary files, it dramatically improves confidence. And quickly, practical point that I would recommend is to use the reporting guidelines that matches your study type. For example, CONSORT for trials, PRISMA for systematic reviews, STROBE for observational studies, and RAI for animal research. This does not just help the journal, but it helps authors to avoid major revision requests. Now, let's make this even more practical. What does good reproducibility actually looks like in a manuscript? In this slide, when we say reproducibility and transparency, the goal is simple. Make it easy for someone else to verify what you did. In practice, good looks like this. The methods are detailed enough that another lab could repeat the experiment without guessing. Key resources are precisely identified so there is no confusion about cell lines, antibodies, or tools. For analysis, especially where computation is involved, journals increasingly expect authors to state the software version and the parameters applied. And for data sharing, the best practice is to provide metadata and explain any restrictions. A common integrity issue is when the claim do not match the data. Reviewers can often sense this when the narrative feels too perfect or when the outcomes are inconsistent across sections. Being transparent about what was planned versus exploratory analysis builds trust. Next, I'll briefly touch on ethics and compliance checks, what journals verify before or during a peer review. In this slide, we explore ethics and compliance checks. Now let's talk about something that often delays decisions, which is ethics and compliance. And I want to frame this very practically. In many cases, papers do not get delayed because the science is weak. They get delayed because key ethical information is missing or unclear. From an editorial perspective at Scifiniti, we often see a predictable set of missing pieces that trigger queries before or during peer review. On the other side, for human studies, the basics must be clearly stated. IRB, Institutional Review Board, approvals, ethics approvals, and informed consent, and where relevant, how privacy and confidentiality were protected. All these need to be accompany the manuscript submitted. For clinical trials, editors commonly check for prospective trial registration, and the registration ID should be stated in manuscript. For animal research, we look for ethics approval plus welfare details. For example, housing, human endpoints, and steps taken to reduce suffering. And for studies involving biosafety or biobanking, authors of any manuscript should confirm permits, material transfer compliance, and proper oversight where applicable. On the journal verification side, editors, like myself, commonly verify conflicts of interest, including financial and non-financial. We also author authorship clarity, ideally with contribution statement such as credit, because authorship dispute are surprisingly common and can become serious post-publication. Another area is data privacy, including data set as anonymized, and is there a lawful basis for sharing or access? And increasingly, we also look for transparency around the use of artificial intelligence tools where journal policy requires disclosure, especially if AI was used in writing, image generation, or analysis workflow. So the takeaway message is ethics and compliance statements are not administrative extras. They are part of research integrity, and getting them right up front is one of the fastest ways to avoid delays and keep your manuscript moving smoothly. Next, I'll move to common integrity issues we see in biomedical and pharma submissions so you can recognize them early before submissions and may avoid them. Now, I want to briefly cover the most common integrity issues we see in biomedical and pharmaceutical submissions. I'll say this upfront, many of these problems are preventable. And in many cases, they happen due to weak documentation or rushed figure preparation, not always intentional misconduct. From a Scifiniti editorial perspective, these are the patterns that most often trigger pre-review queries, reviewer concern, or an integrity check. First is plagiarism or text recycling. This includes high similarity path writing and sometimes salami slicing, where one study is split into multiple minimal papers. Even if the science is valid, this creates trust issues and delays. Second is image manipulation, duplication, splicing, or selective edits. In biomedical research, figures like gels, plots, and microscopy images are common, and even small look-in edits can be interpreted as altering the meaning. The safest approach is keep originals, apply only uniform adjustments, and disclose and splicing or cropping clearly. Third is data inconsistencies. Things like impossible values, mismatched sample sizes across sections, or claims that do not align with the underlying data. A very common trigger is when raw data cannot be provided promptly when requested. We also sometimes see paper mill signals such as template style text, irrelevant citations, or inconsistent affiliations. These are not proofs on their own, but they arise enough concerns that journals may require additional verifications. Then there is the ethics gaps, missing IRBs, consents, missing trial registration when required, or unclear statements about privacy and approvals. These can pause the process immediately because they are compliance issues. Finally, peer review manipulation, for example, suspicious reviewer emails or attempts to influence citations. Journals take this very seriously because it undermines the peer review system itself. The purpose of this slide is not to alarm anyone, it's just to show where delays and rejections often come from. If authors address these areas early, papers move faster and the final publication is stronger. Next slide, I will close with practical editorial expectations, what authors and reviewers can do to prevent these issues and streamline peer review. To close, I want to share practical checklists for authors and reviewers. The things that, from an editorial standpoint as Scifiniti, most consistently help manuscript move smoothly through screening and peer review. First, before submission, run similarity check and make sure every citation is actually relevant. We often see delays because references do not match the claim or because there is batch writing that increases similarity unnecessarily. Fixing this upfront saves time. Second, confirm that your ethics, consent, and registration statements are complete and consistent across the manuscript, especially if you have human participants, clinical data, or animal work. Missing or conflicting statements are one of the fastest ways to trigger an editorial query. Third, prepare figures responsibly, keep the original unprocessed or source files, document any processing, and include essentials like scale bars and clear labels. In biomedical publishing, figure integrity is frequently checked, so good figure hygiene prevents avoidable concerns. Fourth, make your methods reproducible. Reviewers should not need to guess key parameters. Include exact sample sizes and your key statistical decisions. Fifth, align data availability with your claims. If you say data are shared, include the links or the IDs for the data sharing. If access is restricted, explain why and how qualified readers can request it. Finally, if an editor raises an integrity query, respond with the evidence, not just reassurance. Provide raw files, logs, and a clear explanation. When authors respond transparently and quickly, concerns get resolved faster and trust stays intact. So the takeaway is these steps do not just reduce retractions, they reduce review time, reduce revisions, and increase the credibility of your work from day one. Finally, I want to briefly address the role of reviewers because maintaining research integrity is a shared responsibility between the authors, reviewers, and editors. Reviewers play a key role in identifying issues that may affect the reliability or interpretation of a study. The first thing reviewers should check is whether the research question, study design, and conclusions actually align. Sometimes manuscripts overstate their findings or make claims that go beyond what the data support. A careful comparison between the question, methods, results, and conclusions can quickly reveal this. Reviewers should also look closely at figures and images. If anything seems unusual, such as repeated patterns, possible duplications, or unclear image editing, it's appropriate to ask the editor whether the original data or files can be reviewed. Another important area is methods and statistics. Usually, reviewers confirm that the analytical approach is appropriate for the study design and that for the methods are described clearly enough for someone else to understand or reproduce the work. Ethical compliance is also essential. Reviewers should check whether ethics approvals, trial registrations, and conflict of interest disclosures are clearly stated where relevant. Missing information here often signals a need for clarification before publication. In addition, reviewers sometimes notice unusual patterns such as irrelevant citations within the text, inconsistent writing styles, or unusual data patterns. These observations do not automatically mean misconduct, but they are important signals that editors may want to examine further. Finally, the reviewers should try to keep their comments specific and constructive. Instead of simply stating that something seems wrong, it helps to explain what evidence or clarification would resolve this concern. In this way, reviewers help ensure that the final published work is credible, transparent, and scientifically sound, which ultimately benefits both the research community and the integrity of the scientific report. With that, I'll conclude my presentation. I'll be happy to discuss any questions or experiences you may have encountered regarding research integrity during peer review process. Thank you very much for your time. And thank you very much again for giving me this chance to be among you.
Thank you so much, Dr. Muhanad, for that wonderful presentation. And thank you for taking the time to share your insights. And thank you, generally, thank you. Okay, so we are done with the speaker presentations. So you're now welcome to ask questions. In the meantime as well, I'm sharing the link to the Google Doc for the interactive discussion, so you can open the Google Doc and begin filling in your input. As you're doing that, whoever has a question and would like to ask the speakers, Please go ahead. I can see there's a question. I think I'll start with the one that's directed to Miss Noureen. Miss Noureen, there's a question for you. What does your research integrity check involve during your review process? Maybe you could answer that. And then the other one, we will pick who to answer that. Noureen?
Yes, can you please speak again?
The question that's for you is, what does your research integrity check involve during your review process?
Okay, first of all, the reviewers which are assigned to the manuscript will make sure that they have no association with any of the author. Secondly, they are asked to disclose if there is any conflict of interest before accepting any invitation. And then during the review process, they are recommended not to use any AI tool and do not upload manuscript to any AI tool to generate feedback. They may use AI to refine their feedback, but this should be based on their own judgment. And instead of uploading the manuscript directly to any AI tool. Also, the reviewers are asked to maintain the confidentiality of the paper, and they should not cite any portion of the manuscript before it is published.
Okay, great. Thank you so much for that. I hope whoever asked has been answered. And then the other question, I think this one I'll direct it to Dr. Syed. Is the use of AI tools not allowed in medical research or any other aspect of research? Dr. Syed, I'll direct that question to you to answer.
Okay, so the current guidance is not very clear. Even if you look at all the journals, big journals or the new journals, they are not putting up like you cannot use it. You check like BMJ guidance, they have a very whole paragraph on AI. So they're again leaving it to the authors and asking authors to use it responsibly and declare it and verify. And if you have used it to generate anything, so you have to disclose it and include it in your methods clearly. The clearance, their guidance is clear about the authorship. You cannot include AI as an author, but you have to declare and you have to verify all the facts and values or methods. You're taking any assistance from AI, you have to disclose it. But they are not saying that you cannot use it. You cannot find any one journal, peer-reviewed journal saying that you cannot use it. You will not find this information on any journal author's guidance that you cannot use it. So you can use it. Again, you have to declare it. But a lot of time authors, they don't disclose it. And when we detect through our system that somebody have used AI, but they have not declared it, that creates the problem. You are using it and you are not disclosing it. Is dishonesty. I mean, it's not against the transparency and compliance issues. So that create that doubt and the trustworthiness issue. That you are not disclosing it means you're not honest. You have used it and you are not disclosing it. And then the editorial team telling you that you have used your text like 80% from the AI, or you have generated figures, or you have used some AI tools and you have not disclosed it, means you are not honest in disclosing it at the first point. So again, then everything becomes questionable. So general point of view, journals are not clearly stating that you cannot use it. They are just asking authors to declare it honestly, and then also putting this responsibility on authors to verify it, and then you have to then provide enough details about the use of AI. So that is the current status. Even the Scopus few weeks ago, they ask now for all the new the entries. They're asking the publishers to include their policy on AI as well. So they are also not clearly stating that publishers they cannot stop authors to include the AI as as a tool. So there's no clear guidance from the journals or even the databases on that. But they are not stopping you from use. So disclosure and verification.
Thank you, Dr. Syed. I think the next question that was asked, I'll still direct it to you in line to what you were talking about earlier. Pretty much everyone is asking on AI. So this one I'll direct it to you, then the other one I'll pick. How much AI percentage is acceptable for journals during plagiarism check?
Okay, now again, this is a very sensitive and very common question. People ask what percentage. So means there is no criteria. You will not find any clear guidance on the percentage. Even for similarity, there is no percentage which we can say is acceptable. Even similarity check for plagiarism, when we get the percentage, even I see that if even the percentage is sometimes 5%, which is very low, but I see there is a problem with the manuscript because that 5% is taken as it is from somewhere else without proper citation, without rephrasing it. So that's a problem. But sometimes the percentage of similarity is 25%, but I don't see any problem because that is coming from medical abbreviations and protocol and definitions. That's acceptable. So it actually varies. You cannot say that it's we decide based on the percentage. We decide based on where you are using it, how you are using it, for what purpose you are using it, and it's very important whether you are disclosing it or not. So there is no percentage I can tell you that 50% is acceptable or 25% is acceptable. We look at your manuscript and then we decide. We apply then our judgment.
Great, thank you. Thank you. And I totally agree with you. AIs are growing and advancing day by day. So it's all about how do you ethically use the AI tools that exist. And in your research academic life cycle, you're not using it to do everything for you. You're using it as a tool to assist you in doing what you're doing. So I think, thank you, everyone. If anyone has a comment, one last comment before I welcome Ms. Joy to give the closing remarks. Anyone with a comment or one last suggestion or question before I hand over to Ms. Joy? Okay, so if there's none, I'd like to welcome Ms. Joy to give her closing remarks, and then we'll end the session. I'll give a few comments for myself, and then we'll end the session. Welcome, Ms. Joy. Okay, if Ms. Joy is not there, Mr. Harrison, maybe you could speak up on behalf of Ms. Joy. Mr. Harrison? Okay, that's fine. I'll hand it over. But thank you everyone for participating in today's session. Thank you to all the speakers for the insightful thoughts that you've given to our audience. I have learned something, and I hope everyone in the audience has learned something. And this is not the end. We are here to help you, so feel free to interact with us even via email, and talk to us and let us know if there's anything in particular you need to talk to someone about, and we'll be happy to help you. Thank you to all of you for making time to join today's session. And just a few remarks before I close is you'll get the recording for this session before the end of the day. You're going to get a recording of this session before the end of the day, so please look out for that. As well, you'll get the respective links to, Scifiniti, to the other networks represented, that is IAVI, Cambridge, so you can also visit and see what they're doing as well. It also serves as a good opportunity to connect with other organizations who are doing research as well. And also, links to our TCC community and resources that we have available for you as researchers to use. So thank you, everyone, and I look forward to engaging with you in the next webinar. And do have a lovely day. See you on the next session. Thank you, everyone.